ahora tenemos al doctor Joon Kim. Él es un investigador con la posición de Bioinformatics Data Scientist en el Zuckerberg Biohub. Lidera un proyecto para computar redes regulatorias génicas a partir de datos de RNA-seq y ATAC-seq de una sola célula durante el desarrollo del PCB. Estudió también en la regulación de la expresión génica en embriones de drosófila utilizando microscopía de fluorescencia cuantitativa, análisis de imágenes y modelado teórico. Su investigación es apoyada por la Korea Foundation for Advanced Studies. Entonces, le damos la bienvenida al doctor Joon Kim. Mr. Jan, I'm gonna let you know when there are 5 minutes left from your presentation. I'll let you know. So now the floor is yours. You can start up. Thank you very much.
Yeah, thank you, Daniel, for kind introduction, and thank you everyone for inviting me here. It's my great honor to present at this Peruvian Bioinformatics Society today. So I'm Young Jun. I'm a computational biologist at Zuckerberg Biohub, San Francisco. And today I want to introduce what we do at the Biohub in the computational biology platform, and also one vignette where we found some interesting cases of what the multimodal or multiomics modalities bring us in terms of resolving different cases of the cells, and how that can be eased by using some of the interactive tools called cell by gene. So a brief introduction. So as I said, I'm part of this computational biology platform. We have Director Yasin Shembaba and Associate Director Joan Wong. And largely what we do is two things. One, we generate data. We generate really good, high quality single-cell RNA-seq. That's what, here you're seeing some of the flagship projects that we made over the past 5 years. So Tabula is our brand to make the single-cell RNA-seq data sets from across different species. So Tabula murris, Tabula mursinis are both from mouse and different age of the mouse. And Zebra recently published it, recently out as a preprint in bioRxiv, is about the zebrafish during the course of 10 hours to 24 hours of development. And there's also Tabula sapiens, which went into science 2 years ago. It's about human. We also studied how the COVID-19 virus changed the gene expression profiles across different organs by comparing the healthy and COVID patients. And what we do at the computational biology platform is we work across different research groups at the Biohub. There are roughly 10 groups. And we collaborate with them via inventing tools, implementing tools that are already out there, and also with the goal of making this high quality data set and then make them public outside. And if you have any questions, you can add it on the chat and I'll try to check that in time. So before I go into the talk, I want to start with this quick introduction about this tool, Cell by Gene Annotate. So CZ Cell by Gene Annotate is a tool developed from Zuckerberg Initiative, which is our parent organization. It's a graphic user interface-based software for interactive exploration of the single-cell RNA-seq data. And it's freely available. If you go to this website on the address on the right bottom, you can download the tool and you can just upload your own data. You can start annotating and exploring your own data sets. And like I mentioned, the tool itself was developed for transcriptomics, for RNA-seq. But I want to show you today some of the vignettes where how we can expand the tool for other modalities, such as single-cell ATAC-seq or protein or even spatial transcriptomics. And before then, I want to zoom out, start with this statement. And I hope I can convince you that you all agree with this. So when it comes to for us to generate a large-scale single-cell RNA-seq data sets roughly on the scale of 500,000 or 1 million cells, a good cell time annotation was very important. And that process was not very trivial. So how we do it is we usually do our manual annotation of the data based on the marker genes or different signatures in the transcription profiles. Then we cross-check or cross-validate with the tissue or domain experts. And if you ask why do we care so much, then I want to say that good cell time annotation is the first step in any good solid downstream bioinformatic analysis. Because if you want to know about the difference between the gene expression between healthy and COVID patients, here you can think of the red as healthy and the blue as COVID patients, then you'd like to compare the gene expression among the same cell types. For example, if you happen to pick the T cells from the healthy people, and then B cells from the COVID-19 patients, then from gene expression, differential gene expression, what you will get is difference between T cells and B cells rather than difference between healthy and COVID. So ideally, you want to reduce the confounding factors. And to be able to do that, you need good cell type annotation in your data. And there are many dozens of papers out there that does automated or manual methods of cell type annotations, especially in the single-cell RNA-seq data sets. So I want to point you to this nice review paper from Gary Bader's lab at University of Toronto, as well as this tutorial from Fabian Teich's group. But largely the idea is the following. If you have some single-cell RNA-seq data that you can see as gray, if there is some other people's data out there as publicly available that are also annotated, you can transfer the annotation to make the predicted labels on your own data. So the first step would be prediction if there is any reference. And then, but at the end of the day, you will have to do this manual step anyway, because any transfer method, you just cannot just believe them out of the box. So the focus of my talk today will be about this manual annotation, how we can confidently define the cell types, especially using the multiomics. And when it comes to single-cell multiomics, it has been emerging over the past several years. So I think that largely the single-cell genomic revolution started from the single-cell RNA-seq, just because RNA was just more approachable as a molecular modality to measure or capture. And then years after, epigenomics, which can measure the chromatin susceptibility, such as using single-cell attack-seq or cut and run, or even the proteomics, not at the whole proteome scale, but on the scale of hundreds or several hundreds of protein species using the antibody conjugated DNA oligos called ADT. And with these, what do these really bring to the table? So here I'm just showing you some of the well-known technologies such as share-seq or multiome commercialized by 10x Genomics, which can capture RNA and attack-seq. So that gives you both transcriptome and epigenome. And site-seq, which gives you RNA and surface protein information from the same share of cells. And lastly, spatial transcriptomics, which has emerged very rapidly over the last couple of years, which can give you both RNA molecule information and where they are in the tissue. And what does it bring to us, to the biologists? And I think conceptually, what in my mind is like this diagram on the right. So for example, if you have like 3 different cell types in reality, if you have only one modality, what you will see is you might think that they're all from the same cell type, for example, in the projected space of modality 1 and 2, because you just don't see their difference large enough. For example, you can imagine the cells that have very different chromatin stability profiles, but similar gene expression profiles. That's totally doable, and vice versa. So what multiple modalities or multi-omic data gives us is a finer resolution so that you can tear apart different cell types so that you can learn more fine-grained differences along the cell fate determination process. So here I'm showing you some schematic diagram on how we approach this problem as computational biologists. For example, when I interface with the data, it often starts with this on the left side. So I got the cow matrices from, on the rows, there are cells, and on the columns, there are features. And features from the modality 1 can be genes for RNA-seq, and features from modality 2 can be different regions in the chromatin, say peaks for attack-seq, or proteins for the site-seq, or even XY coordinates for the spatial transcriptomics. And how we harmonize these different information from different modalities is oftentimes people compute the UMaps or this dimension with those embeddings for each modality and try to harmonize across different modalities to see which makes sense or not. And I want to show one example where we did that in a very inefficient way. So this was my first project when I joined the bio. So the data set is human PBMC. So it's blood, circulating blood immune cells. So you can think that there are monocytes, macrophages, T cells, B cells, and all those cell types in this data set, this UMAP. And what we measured is both RNA and attack from the same group of cells. So what my first job was annotating this data. So what I did was in the RNA UMAP, I used RNA markers to annotate the cell types roughly. And in the tag UMAP, I also annotated the cell types using the tag markers. And then what was interesting to me is when we projected the tag annotation onto RNA UMAP, it was very noisy. But when we annotated, but when we projected the RNA annotations into a tag UMAP, it was very clean. So something told me that in this specific case, RNA was a better marker to differentiate different cell types than the chromatin sensibility. But for me to actually doing this back and forth in the Jupyter Notebook or any codes in a static setup was very challenging. So only later, like a year afterwards, I found really nice use case of this tool, Cell by Gene, that I just told you, made from CZI. And what this tool enables is, because it doesn't actually care about whether your data set is from RNA or tag, so you can merge your data and then you can move between different embeddings very interactively. So here I'm showing you different list of cell types among the PBMC. And what you can see quickly in a second is you can move to different embeddings. For example, here I'm moving from RNA UMAP to a tag UMAP and project different cell type labels, for example, a tag or RNA, and try to harmonize to see how the cell type annotation that we made is accurate or not. Okay. And this process would have been much harder if we didn't use this type of interactive tools. So we found some potential of this type of tools, and something we wanted to ask was, all right, then having one more modality, let's say having a tag-seq or having protein information on top of RNA, what does it bring to us? What are the things that we can learn? What are the biological insights that we can get? So for example, conceptually, you can think of the clustering or the resolution of the cell types from modality 1 as left and modality 2 as right. In this case, I'm showing you a very conceptual example where in modality 1, for example, RNA, you think that there's only one cell type, but it actually consists of 3 different cell types if you look at the modality 2. And those differences were so subtle in the RNA space, in the transcriptome space, but very apparent in the protein space. And in the next slide, I'm going to show you some of these examples soon. So for example, you can think of these conceptually different scenarios. For example, modality 1 and modality 2 can give you more or less the same resolution of the cell types, or modality 1 can give you only coarse label of the cell types, and whereas modality 2 can resolve those fine differences. And scenario 3 will be more complementary. So the example data set that I want to use today is generated in collaboration with Biohub, Helmholtz University, as well as Yale and Celularity. So this data set takes, again, PBMC, the immune cells, and then the assay that we use is called site-seq. And what it captures is both RNA transcriptome as well as surface proteins. So the way it works is pretty much the same as single RNA-seq done by 10x Genomics. You have 2 different inlets. One, you flow into single cells, and then you flow into beads, and you encapsulate each bead with ideally single cell. And then because you have 2 different types of oligos, one oligo captures RNA, and the other type of oligo captures surface proteins using the conjugated antibodies. And as a result, what you will get is 2 different count matrices. So again, the rows are cells, and the columns can be genes for RNA, the red, or proteins, for the surface proteins as blue. And the first thing that I did was I computed the UMAP for each different modality. So RNA cluster, as you can see on the left. And for protein, I'm going to use ADT, antibody conjugated tags, or antibody-derived tags. So on the protein side, you can also see another UMAP. And because I want you to note that the cell identities from left and right are one-to-one match. So we want to see the relationship between the clusters that we see on the left and right. And that can be easily visualized using a Sankey diagram, which shows you the relationship between the clusters. And for example, here I really focus on this cluster 0, which was the blue cluster on the left in the RNA space. That got split nicely into 3 different clusters on the right. So those were split into this blue, got split into this 3. So what is going on? So we want to go further dive into what are those different clusters in the protein space. And it was interestingly enough, that when we looked at the cell type marker genes or marker proteins for different T cell subtypes, they match exactly to either CD4 T cells or CD8 T cells, or CD56, which marks natural killer cells, which were just seen as one cluster in the RNA space. Of course, solely by using RNA markers, we can still see the separation of these different subtypes of the T cells. But I just wanted to make a point that using protein markers was very easy, much easier than using the RNA markers. Where using RNA markers, you can see that the CD8A and CD4 T cells are a little mixed in this population. So here, one lesson that we learn is, we want to say is that, especially in the case of T cells, where it's very well known by immunologists that the surface protein markers are the most distinguishing factors that differentiate different subtypes of T cells. It's way more straightforward to use a protein signal than the RNA. And one of the piece of bioinformatic analysis that we did is, right, then what does it mean? Does it still hold that protein is better marker or better modality to differentiate cell types? So instead of using the UMass, we went back to the nearest neighbors. So for any single cell analysis, what we computed is for each cell, we ask what are the nearest neighbors in terms of their high dimensional space. For example, in the transcriptome, you can think of these neighbors as having similar transcriptional profiles, and vice versa for the proteins as well. And interestingly enough, what we asked here is that for any CD4 T cell, so here each bar is each single cell, and we're asking how many, what's the fraction of cell types within the nearest neighbors, and the purer it is, the better the cell types are separated in the lower dimensional space. So here, interestingly, what I want you to notice that in here, a lot of T cells, they have CD8 T cells in the CD4 T cell neighborhoods, which means that in the lower dimensional UMAP, it's going to be pretty challenging to separate them out. Whereas in the protein space, it's very clean. It's mostly dominated by CD4 T cells. So CD4 T cell is mostly neighbored or separated out by its own neighbor or own cell types. Where CD8, it also has its own clean neighborhoods. So again, this confirms our hypothesis that protein is just a better marker to differentiate these cell types than RNA, especially in this case. Then you might ask, then what's the correlation between the RNA and protein? Because in here, all the proteins that we have the RNA markers, measurements as well. So we want to see that. And interestingly, when we did the correlation between RNA and protein, here RNA as x-axis and protein on the y-axis, we saw pretty nice correlation for the T cell markers. But interestingly, the correlation between RNA and protein for the same pairs is way higher than the non-pairs, but there's some significant overlap in this lower tail ends. So what that tells us is that RNA is not a good predictor for the protein in those species of proteins, which again validates our hypothesis that, so what are the regime that protein can be better marker than RNA? Our hypothesis is that when the RNA expression is on the lower end, for example, like CD4, then the correlation becomes worse because some interesting features of RNA and protein is that in the RNA-seq, there are just lots of zeros because RNA molecule capture is not 100% efficient. Whereas in the protein, there's another problem. So because we are using antibodies, antibodies have non-specific binding to the cells, even if there's no specific target protein. So you always see these large zeros for the RNA, and you see none of them in the protein, which means there's likely to be some background. So again, coming back to our hypothesis is that, so two working hypotheses. One, RNA and protein are not 100% correlated. So you might want to use RNA for this and protein for that. And two, post-transcription modifications. So even if the RNA expression level is low, the protein's expression step can be another amplification to boost the signal. And you might ask, so then do we just do the protein sequencing instead of RNA? No, then I want to show some counterexample where the RNA modality was better resolving different cell types than the protein. For example, that was the case in this erythroblast population where there's no distinct protein surface markers, whereas the transcriptionally they are very distinct. And for the interest of time, I just want to show you one quick nice vignette application to the spatial transcriptomics, and then we'll end the talk. So the second vignette is about spatial transcriptomics. So as you are well aware, cells don't exist alone in the test tube. So cells in its natural context, it exists in a tissue. For example, here I took some example images from Tabula sapiens paper, where it's showing the villi in the gut. I think. So here, each cells are forming these like each units. So depending on where you sample your cells matters a lot. And largely with the spatial transcriptomics, what people are going to ask is whether the cells that are within the same community are transcriptionally more similar than the others. And those questions can be answered by using some of the new technologies. So it's been several years, but TransVigium is one of the leading first technologies that quantifies the transcript information with the position information within the tissue. So we took one of the sample data sets from an internal study from the mouse brain. So a little bit of background, this is mouse brain with injected tumor. So the mouse is a glioblastoma model where the mouse has healthy brain cells as well as in some neighbors, you should have some tumor regions. And this is the UMAP that we can compute solely from the transcription profiles. And by using some of the marker genes, we can see, I hope you can see like there's cluster 1 and there's cluster 2, visually distinctive in the RNA UMAP. And another nice thing is you can use the browsers to convert those UMAP coordinates into spatial. Now here, what you're looking at is real X and Y axis. And this cell by gene VIP is another tool, a kind of plugin for the cell by gene tool. And here what you can do is you can overlay with the real tissue images, the histology images, H&E staining. So here, each region of this called spot, where the transcription profile was measured, is overlaid on top of where the image was taken. And here I want you to see the clear difference between this dark purple region versus light purple region. So dark purple on the right is showing the tumor region that is injected to the mouse brain. And what I want you to notice is that the tumor is invading the mouse brain here. And it's nice that we captured all these interface between tumor and healthy brain cells. And what was interesting to us is, so here you can zoom in and use the lasso tool using your mouse to curate the annotations based on either transcription profiles or transcription differences. And when you come back to the transcription RNA UMAP, what you see is indeed those two clusters that you see in here are healthy on the right and tumor on the left. And I want you to notice this some small bridge between these two clusters that are in between. And interestingly enough, that those cells that are in between were mapped spatially into exactly on the boundary or interface between tumor and healthy cells. And two explanations. One, this vision method, the diameter of the spatial resolution is 55 micron, which can capture roughly 10 cells each. So those plots that are connecting between healthy and tumor are likely to be a mixture of healthy and tumor cells, or they could be exhibiting like the cell states that are transitioning between tumor or healthy. So that's those two hypotheses that we want to follow up. But if we just use the transcription profiles from those manual annotations, it already gives us some of the nice promising gene cell enrichment pathway analysis. Okay, oops, sorry. So yeah, just want to show the power of these interactive tools when it comes to interfacing with spatial transcriptomics datasets. In that one, you can not only use the transcription profiles to annotate your data, but you can cross-validate using the spatial coordinates or images, which is coming into this official cell by gene product within a couple months. So please be on the lookout. And with these, I just wanted to make a note that cell by gene is not the only tool. There are some variants of the cell by gene family, which is one of them is X-cell by gene developed by my ex-colleague, Alex Roshansky, or cell by gene VIP developed by Biogen, and single cell portal from Broad, and cell annotation platform also from Harvard as well. And oops, sorry. So here I just want to point out that you might ask, so how can we know, or how do we learn how to use these type of tools? And so for that, we put together in a preprint on demonstrating how you can use these interactive tools to annotate different multi-omics data sets. We also incorporated some of the nice slide tutorial that I made in collaboration with the CGI UX designers. So by following those tutorials, hopefully you can see the benefit or use cases of cell by gene or other interactive tools. for exploration of your own data. So with that, I'd like to end the talk, and please be on the lookout of more tutorials coming. And all of the content that I presented today is out on the preprint in bioarchive. You can search for my name or for the tutorial guidelines for manual self-implementation for single-cell multi-omics datasets using interactive software. And lastly, personally, I'm super excited about harnessing the power of multi-omics dataset. And I think we can only do that by being able to interface with them more easily. And I hope that these interactive tools will really enable and bring us to there. So with that, I'd like to thank to the team at the Bio, Alejandro, Angela Pisco. Those were the ones that led this project as principal investigators. There was some help from the CGI single cell team, Meg and Karen, and all like those, they're software engineers or UX designers that helped me with this. And thank you for listening, and I'm happy to take any questions. Thank you.
Yeah, thank you very much, Dr. Jan. It was an interesting talk. So I want to switch to Spanish because the audience is mostly Spanish speakers. Cualquier pregunta que tengan, por favor, pueden escribirla por el chat. Yo voy a hacer el esfuerzo de traducirlo para que el doctor Jan pueda respondernos, ¿no? Okay. First, I'd like to start with some questions of my own. I was interested about the difference between RNA and proteins regarding cell differentiation. You said that proteins were kind of a better fit for that, right? It was one of the previous.
I want to clarify that. So here I'm just I cherry picked this example where the surface proteins were a better marker for different cell subtypes than RNA, which was the T cell or B cells. But also the other case is true where RNA can be a better marker or better modality than protein. As you can see here, there are three distinct clusters, which is showing the trajectory of the normoblast differentiation, which was better resolved by RNA than protein, because in the protein, it thinks that there's only one surface marker, so it's one cell type. So erythroblast, this was better. And another note is when we actually measure this chromatin sensibility using the single cell taxi, that gave us more continuous picture of the cell state transitions.
Using RNA.
Than RNA. Yeah, than even RNA. So, and our hunch was that, in the RNA, you see these different subtypes, like population from pre-erythroblast, erythroblast, and normoblast. But with the taxi, you see more linear trajectory of the cell types, and which gives you more continuum of the cell state changes. And we think that's because in the chromatin sensibility change is more subtle and more gradual than RNA, the gene expression changes at the RNA level.
Yeah, maybe with the evolution of a tumor cell line, that's more of an epigenetic factor, right? Something like interesting.
Right.
Also, I was kind of curious, like maybe with RNA in the previous example, like there was no, it was not that good for filtering cell types. Maybe because there was some background noise in RNA-seq expression. Have you tried different thresholds to filter out these ones?
So in the RNA, actually, the error or the noise is mostly coming from the dropouts, because by dropout, what I'm saying is not being able to detect the RNA molecules that are present. For example, we are losing a lot of like rare, rarely expressed like genes in our count matrices, whereas in the proteins, the other way. We always detect something. We just don't know whether the level is like true or not. So we use pretty stringent threshold to filter out those. I think in the end, it's a little subjective, like which threshold you pick and how you filter them out. But largely, I would recommend just for each gene or across different genes, I recommend you to like see the distribution of them along like histogram or something like that.
Okay. Just to check out this.
Yeah.
Oh, excellent. Well, I believe there are more questions. Chicos, cualquier pregunta, por favor, que le escriban ahorita. O siéntanse libres de prender su micro y poder preguntar directamente. Sí. They are typing. Hay una pregunta acá de Diego Taquí. Adelante, Diego.
Yes, thank you for the talk. It was very interesting. I wanted to ask For example, I see that multimodality data like single cell or a taxi is included in this cell to or cell X gene repository. But I have also seen like this advance in machine learning algorithms to predict annotation of single cell RNA sequence data, different subtypes. For example, I remember one's name, single cell GPT, published in Nature Methods, and there is a lot of them. Right? So is there, what do you think, is there any plan to include this kind of annotation by machine learning, or do you think it's still not yet as good as, for example, other fields like AlphaFold prediction that are very, very, very good? What do you think?
Yeah, thanks for the question. Great question. So I think it's mostly about this. So I think you're right, and it's a bit of chicken and eggs or chicken or eggs problem because all the single cell GPT or those foundation models based on single cell RNA-seq or single cell omics data were actually trained based on these large scale corpus of data, and they all use the cell biogen census, which hosts like 88 or 70 or 30 million data sets at the time. So to make a really good machine learning model, you need good quality, nicely annotated data. And at the Biohub, our job was making this data, nicely annotated data. And these data enable making those first round of foundation models, which will hopefully give us like really accurate prediction of the cell type labels for the newly generated data. I hope that's coming. I think that's coming. But when it comes to like in the lab or in any research projects, you will definitely start with making those predictions using those models, and there are many out there. Not only those foundation models, but also SCVI, Harmony, Sociedad, all those tools that predict the labels using the reference. That's step number one, I would say. But in the end, you will have to like look at your data on your own. And I think in that sense, it's not only limited by annotating cell types, but it also means using these interactive tools, you can actually see like what's the difference, what's the subtle difference between the data that you have.
Okay, yeah, that's very interesting because I just remember that these giant models are training this same resource, right? And
Right.
I guess you could say that the advantage is for the well type of cell type that are not as represented in this kind of data sets. But again, if the data set does not have this data to start with, maybe it is not as good, right? But yeah, thanks, very interesting. I am actually part of a journal club project in this Society of Bioinformatics in Peru.
Yep.
So we will actually love to have you someday for our journal club also. So yeah.
Yeah. Thank you. Thanks for the great questions.
Genial, amigo. Muchas gracias. There's one more question, Mr. Jan, which is, let me read it out for you. In the study of brain tumor model in mice, how are spatial and RNA modalities integrated to analyze the interface between tumor tissue and healthy tissue? And what advantages does this integration offer for understanding tumor progression?
Oh, I see. So in this specific case, we had like tumor that we can clearly see from our eyes from the H&E stain images. And because we took the Visium data, so for the first part of the question, how did we do the integration? We actually took the separate single-cell RNA-seq data from the different slices of the brain, which you could annotate well by like healthy and tumor and also different cell types for the brain. So you could see like different astrocytes or macrophages or different brain or neuron cell types. So actually with these, you can subset and annotate different subtypes well for the healthy parts. But for the spatial, because as I mentioned, Visium captures like roughly 10 different cells. So here each dot is not a single cell, it's a collection or clump of cells. So it's actually more challenging to see these different sub-cell types in the Visium data. I think there's some limitation in the assay itself. So that's why we didn't like dig deeper into like, transferring those labels from single-cell RNA-seq to the Visium. We tried that, but in spatial transcriptomics such as Visium, what people normally do is not actually interpreting each spot as a cell type, but more like computing the fraction of cell types within each spot, which is called deconvolution. So we tried that. The result was okay. It wasn't like necessarily more interesting or less interesting. But something we want to really follow up is on the direction of, so what's going on at this interface between tumor and healthy cells? And like what are the cell type compositions in this regime, like which can be mapped into right in the interface between tumor and healthy? And if we use more fine-grained spatial resolution methods such as Fly-seq or Murfish, commercialized by Vision, what are the signatures of gene expression that we can see between those interface cells? Which we didn't have a chance to follow up, but yeah.
Yeah, thank you. Thank you very much, Dr. Jan. It was really interesting. I saw that the project kind of was open on GitHub, right?
Yeah, yeah.
So the codes are mainly, the plots are mainly created with R and Python, or are there any other languages involved in the development of this?
So we mostly use Python, and in the GitHub repo associated with this preprint, we put together all the Jupyter notebooks and all the Python scripts that are used to generate all the figures that go into the paper. So that's reproducible, and hopefully users can take advantage of those for any standardized like multi-omics analysis. When it comes to single cell ATAC-seq, I just have to mention that I think still the best tools out there are in R, not Python. So for that, we had to use R. But other than that, everything is Python.
Yes, process transcriptomic data. There are bioconductor packages, very useful ones.
Yeah.
Well, I believe that's all the questions that we have for now. Thank you very much, Dr. Jan. I'd like to extend the invitation for further collaboration with the Peruvian Society of Bioinformatics, any journal clubs or any other talks about tools that created in the Zuckerberg Biohub. We were very pleased with your presentation. It was really exciting. Thank you very much. Hope to see you soon.
Thank you. Thanks for the invite, and thanks for listening. Bye.